All right, so today I'm going to talk about evapotranspiration of plants' water use on landscapes. So basically, this is the title of my talk, Evapotranspiration in a Subtropical Wetland Savanna, using low-cost lysimeter, eddy covariance, and modeling approaches. So briefly, evapotranspiration is the sum of soil evaporation and plant transpiration. Okay? It is the largest water loss from subtropical and tropical ecosystems, and it is almost equal to rainfall in these parts. And I'm going to refer to evapotranspiration as ET. Okay. All right. So the challenge is that there is no way to measure ET at a landscape level. There are methods such as eddy covariance and vapor transport models and remote sensing data sets, but there is a lot of uncertainty in these methods. And much of the world actually has unverified ET data, which in fact, in turn affects water resources management, and drought forecasting, and understanding the effects of climate change and land use change on ecosystems and water availability. As an example, this is a map of an eastern part of Tanzania where I worked before. So we have MODIS remote sensing data, which is pretty good spatially. Like the green ones are the mountains. So we have higher ET in the mountains and in the river valleys, but in the drier areas, we have less ET. So the spatial representation of MODIS as satellite-based ET data is good, but the values, the actual magnitudes are way off, and sometimes it's twice the amount of rainfall, for instance. So, and one reason for that is that these data sets in much of the world are validated using the network of eddy flux stations, but which are mainly concentrated in North America and East Asia and Europe. Africa, for instance, South America have very few. So the objective of this study was to design two low-cost lysimeters, a weighing type and a water level type, and compare the data from these against an eddy covariance tower we have on site at Bakalan Ranch. Again, using commonly ET models with local weather data and ET data for the region. And the idea is that if these low-cost lysimeters provide ET estimates or measurements which are similar in magnitude and seasonality as other methods, then these can be easily achievable independent checks in these remote parts of the world where there is no ET data. So the study site for seeing how these low-cost lysimeters work was Bakalan Ranch, which, as we know, is the dominant land use is basically ranchlands. And the land cover is grasslands, wetlands, woodlands, and there's irrigated agriculture. Just some pictures showing the different land use types in Bakalan Ranch, pastures, wetlands primarily. So the first type of lysimeter we designed was the weighing type lysimeter. It's a pot that is water to fill capacity and weighed. And then a few days later, it's weighed again. And the difference in, weighing in the weight, that is, wait a minute. Sorry about that. Yeah, so the difference in the weight, I'm trying to get this out of the place. Okay, so the difference in weight in these two days is basically the ET, right? So we had 20 lysimeters, and in each of these, we had a single species. And the species chosen were, they occurred in Bakalan Ranch across grasslands and wetlands. And they were the Axolotl fucifolius, which is a grass, a forb Phalanodiflora, Paspalum muratum, Bahia grass, and a sedge, Rhynchospora colorata. The second type of lysimeter we designed was the water level lysimeter, where basically this is a mesocosm. It's a big plastic tank where the water level is added to a certain mark. And a week later, the drop in water level is observed. And the difference in the water levels, you should calculate the volume of water, that is equal to the loss of ET on the landscape. So this is an example in our shade house. These are our water level lysimeters in the background, and these are our weighing lysimeters in the foreground. Now, to compare these two methods against independent ET estimates, we had Eddy covariance. We have a tower on site, which is run by the University of Illinois, and we use the latent heat flux methods to obtain Eddy covariance. We also used weather data from our weather station to run various evapotransport models, FAO Penman-Monteith model, modified Turk, and adaptive simple variation model. And lastly, we obtained ET data from the Florida Automated Weather Network. The results. So this is looking at annual ET in millimeters. On the left side, we have the 4 weighing lysimeters for the 4 different species. The 5th bar is the water level lysimeters. Okay, and that's basically an average of 30 lysimeters, which we had mixed plant communities. On the right side, we have the 3 ET models. Then this is the ET data for the region, and this is rainfall. So as you can see, they're pretty similar in a way, the same order of magnitude. This is the monthly ET now. So in the gray bars, we have the monthly rainfall. The orange line shows the ET from the Eddy covariance tower. Then the top 3 lines are the monthly ET for the 3 ET models which were run. And the purple line is the ET for the data. You can see that in the summertime, the growing season, you have high ET. So that's the expected seasonal pattern. So these were again the models. Right? Now looking into our lysimeters, this is again monthly data for the lysimeters. The blue line is the average for the water level lysimeters. Again, there are 30 of them. It shows the same pattern where as the summer comes along, the ET increases and then it drops. The bars are the weighing lysimeters. So each bar, like the green bar here, for instance, is the ET shown by the sedge. So to look at them side by side now, the monthly, the y-axis scales are the same. So first thing one can see is that the magnitudes shown by the lysimeters are pretty similar to what is shown by the models, especially the ET data for the region. And then they all show increasing ET over the summer season. And interestingly, in the month of May, which normally has the highest ET for South Florida, we had a drop. And the reason for that was May was extremely cloudy. And so what the lysimeters are showing is also being picked up by the models as well as the Eddy covariance. So that was an interesting sight. So to discuss this, so we said that both types of lysimeters, the weighing lysimeters and the water level lysimeters, they gave ET values which were similar in magnitude to the models and the data for the region. And then the lysimeter ET values are slightly lower than the ET models, and that is to be expected because the ET models are calculating potential ET, that is the amount of water loss from a landscape if there is no water limitation, like for irrigated agriculture. And then both the lysimeters and the models, as well as Eddy covariance, showed an unseasonal drop in May, which I spoke to before, because May was just so rainy and cloudy. And as we know, that plant transpiration decreases during cloudy conditions. So this is now the other thing we saw, that these the weighing lysimeters were also sensitive to different species effects. For instance, Rhynchospora colorata, the sedge, because it grows in wetland margins, it's probably, it is found to have the highest ET. That's the green bar on the left. So the highest ET was shown by the sedge, and the lowest ET was shown by Axonopus fucifolius, which is the yellow bar on the right. And it's possible that, so basically these lysimeters are sensitive to show different amounts of transpiration and ET happening for different plant species. The other 2 species had intermediate effects. And it's possible that, say, plants which grow on the margins of wetlands, they might be transpiring more because usually they face less water shortage than, say, upland grasses, for instance. This is just, but again, these results were based upon 5 lysimeters. So we had 5 lysimeters for each species. So n is equal to 5. And it could be an interesting study to extend this, to other species. Okay. Some of the considerations in using these lysimeters. The ET values we get from these lysimeters are in between potential ET and field conditions because these were artificially watered. The weighing lysimeters were watered once in 3 days, and the water level was watered once a week. So there were times when the lysimeters dried out and had possibly water stress, but the watering was not the same as actual field conditions of rainfall because these were in a shade house with a plastic roof, which basically cut out rainfall. The weighing type lysimeters can also be subject to operator error, basically because initially we watered the plants to saturation and then to field capacity, and then we weigh it. So if somebody were to wait for a lesser time, then the weights could be different. Also, because these plants are grown in pots, their roots are constrained to a certain zone, and so therefore there can be edge effects. So these are some considerations to be used while using these lysimeters. So to conclude, both weighing type and water level lysimeters can be used to calibrate regional ET models for improving their accuracy, especially in large parts of the world where we don't have actual field ET data obtained from Eddy covariance or other methods. So also that, these lysimeters can be a good way to actually measure the loss of water from mixed species communities, which typically occur in the landscape. With that, I'd like to end this brief talk. Any questions? Let me know. Thanks.
Thank you very much, Amartya. Does anybody have any questions? I don't have any currently in the Q&A. Oh, never mind, I think. Hillary Swain has one. She asks, why is the UFL fan network model more close to the local Lysimeter measurements? And what is the potential ET model? Or what is their potential ET model, excuse me?
Right, so the ET data from the FAWN network, let's see, I can show it on this one. So the FAWN network, basically, it uses the FAO-Penman-Montieth equation, which calculates potential ET, but then they calibrate that with an eddy flux network across Florida. So that's the reason why I think the FAWN ET data is less than the potential ET models. But again, it's higher than the Eddy covariance data. And it's been, a lot of researchers have found that ET obtained from Eddy covariance is typically lower than ET obtained by other methods, such as commercial lysimeters in agriculture. And reasons given for that are that eddy flux towers have energy balance closure issues, and plus there are issues about how representative is the footprint for the vegetation. So, but yes, I mean, so I think, and the encouraging thing was that, so if you look at the annual ET, this one here, ET FAWN, that's the Florida Automated Weather Network ET data. And these 5 bars on the left, these are the ET measurements obtained by our lysimeters. The 4 were for different species. That's for the sedge, that's for the grass, and this was for the mixed communities in the water level. So on an annual scale, that the measurements or the values obtained from these lysimeters were kind of similar in the same order of magnitude as the ET, from the Florida Automated Weather Network data.